Hey guys, Sriram Biology from Asia Express back with another biology video. Today we're going to be solving 2 May June 2024, so it's an MCQ paper. Now the theory and AT we are done for the October-November 2024 session. We're going to move on to the MCQ paper as it's the last paper for extended. So let's get straight into it. So first, which process carried out by all organisms? So your teachers might have taught you the Mrs. Gren. So Mrs. Gren, and just fit that in. Yeah, so Mrs. Gren, these are all the characteristics of all living organisms. So that stands for, as we go from left to right, movement, respiration, sensitivity, growth, reproduction, excretion, and nutrition. So we can easily cut it out. So for transpiration here, that only occurs in plants, not in other organisms, so we can cut that out. Sexual reproduction, there are organisms that reproduce by asexual reproduction as well, so we can't classify it like that. Photosynthesis, again, only by plants. Finally, growth. Growth is the right answer. It's the letter G here, so we're just gonna go ahead and tick that. Now, what is the first part of the scientific name of organism using the binomial nomenclature system? So in the binomial system, we have genus first, genus, and then you have species. So it's genus first, so that straight away gives us answer 2A. Let's go down. So in which labeled part of the cell, as a plant cell, is glucose made? So let's go from A to D. So vacuole usually just stores cell sap, it doesn't produce glucose, so that's wrong. B is actually the right answer here, chloroplast, because chloroplast is the site of photosynthesis. It contains chlorophyll, and so it does, chloroplast is where photosynthesis occurs, and glucose is produced via photosynthesis. So from that we can get our answer, so it's B. D, cell wall, is just to provide shape for the cell and just for rigidity of the cell. Finally, you have nucleus. Nucleus doesn't produce glucose because it just controls cell function. It doesn't actually produce anything like glucose. So B, A, B. Next, let's move on to the fourth question, magnification. So the distance between X and Y. So on the diagram, so that tells us that this is image size. You're going to understand why I'm making this classification soon, but that is image size. Then the actual length of the cell between X and Y is 150 micrometers. This is actual size as it's given in the question and the statement. So that's actual. So magnification is given by image size divided by the actual size. So they're both in different units, so you need to convert them to this common unit. So you can either convert 60 micrometers multiplied by 1000 to get the micrometers, or you can divide 150 micrometers by 1000 to get the value in millimeters. I'm just going to divide 150 by 1000, so I get 0.15 millimeters. So 0.15 millimeters, and then 60 divided by 0.15 millimeters, so it's 60 divided by 0.15 millimeters, so that gives us 400. That's the answer for C. Okay, now some plant cells are placed in distilled water for an hour. What is the description of the plant cells after 1 hour is correct? So here we have to think about osmosis. So osmosis is the movement of water molecules from a region of higher water potential to a region of lower water potential, down a water potential gradient across a partially permeable membrane. So here distilled water has higher water potential. Just put higher water potential. You always have to remember that distilled water, higher water potential, and plant cells are lower water potential. So by osmosis, water is going to move into the plant cell. But now we have these terms, important terms here. So burst does not apply because plant cells do not burst because they have a cell wall. Flaccid and plasmolized also don't apply because that only occurs when water moves out of the plant cell. That too, plasmolized. Yeah, so plasmolized and flaccid don't apply for when water moves into the plant cell, only out. So that leaves us with turgid. Turgid is the correct term for plant cell. Okay, so which diagrams represent active transport. So active transport, it is the movement of molecules from a region of lower concentration to a region of higher concentration against a concentration gradient using energy from respiration. So it's across partially permeable membrane. But in my description, you heard me say using energy from respiration. That itself allows us to cut out the options C and D, because they are saying it does not require energy from respiration. And then you're left with A and B. A, it shows it actually going from higher concentration down to a lower concentration, which is actually not what we are looking for, because we need the opposite, lower concentration to higher concentration. So A is wrong, leaving us with B. Okay, now the diagram shows bases in a section of DNA from one strand only. So the complementary DNA strands. The complementary DNA strand is going to have the complementary nitrogenous bases. So as you already know, adenine pairs with thymine and cytosine pairs with guanine. So you're going to look for a base pairing that starts with TG, because adenine has to be, so there has to be a T here and a G there, because that's how the nitrogenous base pairs work. So GC doesn't make sense, because it has to start with these two, T and G. So GC, GT, TC, all three don't make sense. That leaves us with D, 7D. 7D. Okay, so now we have the effect of pH on the rate of an enzyme catalyzed reaction. So firstly, pH increases from 5 to 7, the kinetic energy. So kinetic energy, we cannot use the explanation of kinetic energy when it comes to pH, because kinetic energy applies only when we have the temperature changing, not the pH. So as the pH increases 7 to 9, frequency of, okay, so B is also wrong, because from 7 to 9 it's actually going down, the rate of reaction. The description they have given is when when the rate of reaction increases. So here that doesn't apply. So that's also wrong. And the shape, okay, and then the D option, substrate denatures at pH 7 is also wrong, because we can see that is where it's at its maximum rate of reaction. So that's also not true, leaving us with C. The shape of the enzyme's active site changes as pH increases from 7 to 9, because this is actually true. So this is how denaturing works. So active site is where the substrate is going to fit in and how the enzyme actually breaks down the substrate. But we can see the rate of reaction is decreasing. So obviously this is because the active site shape is changing. So the substrate no longer fits in and the rate of reaction won't increase. So the option is C. Let's keep moving on. So which sentence explains the importance of vascular bundles for photosynthesis? Okay, so firstly, vascular bundles transport carbon dioxide to the leaf. That is not true. That is done by stomata. Vascular bundles are phloem and xylem together. Phloem transports substances such as sucrose or amino acids and other such assimilates, whereas xylem transports dissolved minerals and water. So those are the only things it will carry. They don't transport oxygen. Again, stomata will help with that. They do transport water to the leaf. That is true. And transport starch to the leaf? No, it doesn't transport starch. Leaves, you have to differentiate. They transport sugars as sucrose and they store sugars as starch, and they produce sugars as glucose. So actually they only transport sucrose, and that too not to the leaf most of the time, but that depends on the season, etc. Here we're not going to look too deep into it. D is the obvious right answer, so let's just go ahead with D. Which substance is the product of protein digestion? So here we have to think about the monomers. So proteins are made out of amino acids, so straight away we can have A. Fatty acids, glucose is for polysaccharides, glycerol and fatty acids are for lipids, so we don't have that. It's straight away amino acids, the obvious answer. So which organ produces amylase? So this helps break down sugars, amylase. So this is produced in esophagus, doesn't produce any such enzymes. Kidney also no. Gallbladder doesn't produce amylase, produces other such enzymes. Usually it produces neutralizing enzymes and also enzymes that break down mostly lipids and fats. 11, the answer is D. Pancreas produces amylase. This is a question you're going to see in many papers. So which substance is absorbed by the lacteals in villi? So the villi, you need to know the structure of villi. So you have the drawing like this, with many capillaries inside and then microvilli of the surface. So this drawing is very inaccurate, guys, but there's a capillary coming in. It's going to get intertwined. It's going to go out. Blood comes like that and goes back like that. So you have a certain thing called lacteals within this. So each part absorbs a different thing. So all of actually lactic acid and hydrochloric acid are wrong, but amino acids and fatty acids are absorbed by the villi. But they are asking specifically for the lacteal. Lacteals always only absorb fatty acids. So we're just gonna go 12b. So 13 question. The alimentary canal. Where is most water absorbed? Okay, so let's just go ahead with this. So first let's label it. So A is the liver, B is the stomach, C is large, I'm just gonna write large intestine, and this is small intestine. So the large intestine is on the outside and the small intestine is coiled up within it. So the liver and stomach don't do any absorption. So straight away we cancel A and B. C, the large intestine only absorbs the remaining water and what it requires in smaller amounts. D is what does most of the absorption of water. So the answer is 13D, the small intestine. Okay. So here is the villi structure I was talking about earlier, where you have the capillary with the blood flow, lacteal absorbing fatty acids, and then you have other cells here. So on which cell surface are microvilli found? So microvilli, they will be present on the most exterior or the outermost layer to increase surface area of absorption of the nutrients. So here it's not going to be on any of the inner layers, so we can straight away cancel that. The answer is just D. Because it's on the outermost layer, we can straight away just conclude that the answer is D. So which feature of xylem provides support to plants? 15th question. So support to plants is actually the key term here. They're trying to confuse us. So first of all, A is wrong either way. Cells are joined. No, that is actually right. Sorry, it is correct. But keep in mind the thing: support to plants. A, B, and C are all adaptations of the xylem to transport water, but none of them are adaptations to provide support to plants. Only this point here, lignin being present, is the fact that helps provide support to plants, because lignin helps keep the xylem strong and allows the plant to remain upright. So that's why we need to remember that point. Don't get confused by the fact we're given adaptations. We only need to see what helps the plant. Okay. So which change in an environmental factor will increase the rate of transpiration? So here these are the 4 factors that mainly affect transpiration. So decreasing light intensity is actually going to decrease the rate of photosynthesis. So this actually leads somewhat like a chain of processes. So decreasing light intensity decreases rate of photosynthesis. So the stomata is going to open less. Why, you may ask, is because when more photosynthesis occurs, you're going to need more carbon dioxide to enter and more of oxygen to exit. And this gas exchange is done by the stomata. But when you have lower photosynthesis, the gas exchange, the rate of gas exchange is going to decrease. So the stomata is going to open less, and in transpiration, the evaporated water has to exit via the stomata. So with decreased light intensity, decreased stomatal opening, so decreased transpiration. Decreasing temperature, again, temperature increases the kinetic energy of the water molecules and it allows them to evaporate easier and faster. But when there's a decreasing temperature, they have lower kinetic energy. The water molecules are going to have lower kinetic energy. So when you decrease temperature, the rate of transpiration will decrease. Finally, the wind speed. Wind speed provides draught. You might have learned this in physics. So the higher wind speed, there's a higher draught. And so from the surface of the water molecules will be taken and the rate of evaporation will increase when the wind speed increases. So with the decreasing wind speed, again, the wind speed with the rate of evaporation, rate of transpiration will decrease. Finally, decreasing humidity is the final answer. There's also a direct explanation for why it's decreasing humidity. So humidity is basically the amount of water content you have in the surrounding atmosphere. So transpiration works on the basis of osmosis, essentially. So when there's a higher humidity, the atmosphere surrounding the leaf has a higher water potential. So the water potential gradient is less steep, and so essentially there'll be less transpiration when there's a higher humidity. But when there's a decreasing humidity, the rate of transpiration is going to increase because the inside of the leaf will have a higher water potential and the outside will have a comparatively lower water potential. So rate of transpiration will increase. 17. So 17, what describes translocation? Okay, so translocation only occurs in the phloem, like of assimilates, like I was mentioning before, amino acids and sucrose. It's not going to happen in the xylem. So straight away B and D are wrong. So the movement of amino acids and sucrose from the source to the sink in the phloem. This is the right answer, A. And C is the wrong answer here because it's saying sink to the source. Source is always where it's coming from. Sink is where it's going to. So you can't have it move from the sink to the source, only from the source to the sink. So 17A. Okay, which pathway is taken by blood in a fish? So fish have a single circulatory system. It only passes through the heart once through a complete circulation of the body. So how is it going to move? So first, gills and body. So straight away we can cancel out A and B because it needs to start from the heart. You can't start from the gills. It's gas exchange. It's for breathing. It's not for where the blood starts from. So that can't be right. So heart, body, gills, body. So that it's not really right because it suggests that it's going straight from the gills to the rest of the body, and if it even wasn't like that, it's suggesting that's not right. So the first is saying it's from the heart to the body, then to the gills to the body. That suggests a double circulatory system, and that doesn't apply for fish. So it has to be C, heart, gills, body, heart. So it's a single circulation. So you can see, so when it goes from heart, gills, body, only then the heart. It only passes through the heart once in a complete circulation. Okay, so which component of blood produces antibodies? So plasma is a component of blood, but it doesn't produce antibodies. It mainly just contains dissolved nutrients and enzymes, etc. So it's not A. Platelets, they help form scabs and a physical barrier to pathogens and infection. So example, when you get a cut, it helps you form a scab then, but it doesn't help you produce antibodies. Red blood cells, they just carry oxygen binded with hemoglobin. They just transport oxygen throughout the body. That's why we're left with the white blood cells, which have lymphocytes and phagocytes, where the lymphocytes are going to be producing antibodies specific to the antigens on the pathogen. So that straight away we can get the answer 19D if you want a more direct approach. So the cholera bacterium produces a toxin which affects humans. What is an effect on this toxin on the human body? Okay, so secretion of chloride ions from the blood into the lumen of the large intestine. So that's not right because this cholera bacterium affects the small intestine. So large intestine is wrong, and it's into the small intestine, into the lumen of the small intestine, not into the blood. So again, C and D is also wrong. So that just leaves us with B. But if you want a more direct approach, if you know how cholera bacterium works, essentially the cholera bacterium, it's going to secrete chloride ions from the blood into the lumen of the small intestine, just like the answer 20B. So what this does is this decreases the water potential in the lumen of the small intestine. So when this happens, more water is going to enter the small intestine, and it's going to make your feces more watery, and that's what results in diarrhea. So 21. Next question. These changes occur in the human body during exercise. So the breathing rate increases, rate of respiration increases, nerve impulses sent from the brain to the intercostal muscles and diaphragm. Receptors in the brain detect increased concentration of carbon dioxide in the blood. Okay, so breathing rate increases and rate of respiration increase. They're asking the correct order. So breathing rate increase and rate of respiration increasing. This cannot be in the first two stages. This cannot be the beginning. Either of these can't be the beginning. So you can cut out anything. Rate of respiration starting is actually right. Very sorry. Rate of respiration can start, but anything starting with 1 is wrong. So you can't have it like that. And so because only after 2, 3 and 4 occur, 1 can occur. 1 has to be the last. So that itself gives us the answer D. But let me just explain it in greater detail. So first, yes, the rate of respiration will increase because you need to oxygenate your blood faster because your muscles are contracting, they are working, and they need oxygen to respire, so their rate of respiration is going to increase. So starting with 2, the number 2 being the first step is right. But then what happens is there's an increased concentration of carbon dioxide in the blood, the carbonic acid, decreasing the pH. So chemoreceptors in your brain, the specific word is a chemoreceptor. So this is going to detect this increased concentration of carbon dioxide in the blood. And why? Because I just mentioned the muscle cells working, they're respiring, so they're releasing carbon dioxide as a result, and that is going to increase concentration of carbon dioxide in the blood, and that's going to be detected by the chemoreceptors in the brain. Then after 4, you have the step 3, because after it detects, only the nerve impulse will be sent. So that's, as a result, you have 3. So the order is, this is step 1, this is step 2, this is step 3, 3 is step 3, and 1 is step 4. So this is step 4, and this is step 3. So it's 2, 4, 3, 1. That's the entire process. So after all, the breathing rate will increase to get rid of the carbon dioxide. Okay, so 2 molecules of glucose are aerobically respired. So aerobic respiration is a breakdown of glucose using oxygen to release energy. So the formula goes like this. So it's, I don't know if I'll be able to write it all out. So C6H12O6 plus 6O2 gives 6CO2 plus 6H2O. So 6H2O is what's important here, and that's with 1 molecule of glucose. Now it's 2, so I'm just going to multiply this by 2 as well, so that leaves us with 22. Sorry, just draw it a bit better. 22D. Now let's keep moving on. Which substance has a higher concentration in the renal vein than in the renal artery? Okay, so in the renal vein, carbon dioxide, glucose, and everything else is not actually it's not going to be higher concentration in the renal vein than the renal artery. So we can straight away cancel all that out. Glucose, oxygen, no. Urea will go through to the bladder and be excreted through urine. So carbon dioxide is always going to be the right answer, because the veins, except the pulmonary artery and pulmonary vein, those are the exceptions. The veins are always going to have a higher concentration of carbon dioxide, so it's 23A. Where are amino acids deaminated and converted into urea? Here there's not really an explanation. So actually it's taken to the liver, and that's where the process is done. It's assimilated in the liver. So it's 24C. You just need to know that organ. You need to remember that. Which process moves neurotransmitters across a synapse? Here it's not a gravity. First of all, it's not a process really. And also it's just not how it works. It's not gravity. Osmosis, these are not water molecules, so osmosis can't occur. It's moving from a higher concentration to a lower concentration, so it's not active transport either. So that leaves us with diffusion. When the blood glucose concentration is low, which hormone is released and which organ releases it? So how blood glucose concentration, this is a negative feedback or the homeostasis process. So when blood glucose concentration is low, glucagon is released. And both insulin and glucagon, both which help maintain our blood glucose levels, both are released by the pancreas. So this answer has to be 26B. Okay, a shoot grows towards the direction of light. So let's just have a little sun here. Yes, very bad drawing, I know, but I'm just gonna have to manage. Okay, so cells on the side in the light have more oxygen. So that is not true actually, because the oxygen is going to gather on the side with more shade. So if this is the side with shade, what I'm marking here, then the oxygen will gather here in larger numbers. So 1 is wrong, 2 is right. Cells on the side in the light elongate more. No, it's cells on the side in the shade that elongate more. So 2 and 4 are right, and 1 and 3 are wrong. So straight away anything with 1 or 3 is wrong. So this is wrong, that's wrong, this is wrong. and 27 Ds, right? So if you want a more direct explanation again, so when the sun is shining, the side with the light, near the light, is actually going to have less oxygen. The oxygen is going to diffuse to the side with lower light, the side with shade. It's going to cause cell division and elongation there to cause the oxygen tip to bend towards the sunlight. So that's what gives us the answer 27D. Okay, so which statement about antibiotics is correct? So antibiotics are used to treat all transmissible diseases. No, that is not true. Transmissible diseases are true, not all of them. They are used to treat diseases caused by viruses. No, they are used to treat diseases caused by bacteria. That's why Biotech is there. So antibiotic resistance can be stopped by increasing their use. No. If you know about the example of Staphylococcus aureus, sorry, I forgot the exact name. So Staphylococcus aureus, when you use more antibiotics, that's when that's why the bacteria build resistance. So you can't actually decrease antibiotic resistance by increasing the use. In fact, you're going to increase antibiotic resistance by increasing the use. So that's also wrong, leaving us with 28D. Okay, so Fusarium wilt is a fungal disease of bananas that can spread quickly through a banana population. The table shows the area affected by Fusarium wilt over time in one country. So 1976 to 1999. So percentage increase, so that's just new value minus the original value divided by the original value into hundreds. So let me just write that out here. So that's going to be 3000 minus 1200 divided by 1200. So let me just type that into my calculator here, and you get 150%. 20. That's the answer is 29C. We move on to the next one. So the diagram shows parts of a flower. So which processes have taken place? So here we can see the pollen grains at the top. Those are pollen. So it has grown its way, so the micropyle has grown its way, the pollen tube has grown its way down the style all the way into the ovule, and now it's about to fertilize. It hasn't happened yet. So pollination has definitely happened. We can see the pollen there. So it's either 27C or D. These two are wrong because pollination has evidently occurred. Fertilization hasn't happened yet. So it's 30C because fertilization hasn't happened yet, but pollination has. Okay, the diagram shows part of a placenta and umbilical cord. What is the function of the umbilical vein here? So umbilical vein, we can clearly see the blood is flowing towards the fetus. We can just write fetus here. So we can clearly see this is the mother's blood. So the umbilical artery is taking blood from the fetus towards the mother, and so gas exchange and diffusion, etc., can happen here. So if it's going towards the fetus, so first of all, umbilical vein and the veins and arteries aren't involved in the pathogen, toxins, and those. It's not involved in those areas. It's mainly involved in gas exchange and nutrient exchange. So 31C, we can clearly say it's taking blood towards the fetus. So we can't say it's transporting carbon dioxide away from the fetus because it's evidently taking blood towards the fetus. So the answer is 31D. It's transporting oxygen towards the fetus. Let's keep moving to the next question. Okay, so these kind of questions are sometimes a bit confusing. So the graph shows how the mass of DNA per cell changes before, during, and after a nuclear division in a diploid cell. So, okay. Which row describes the type of nuclear division, the chromosome number of the daughter cells shown in the graph? So it is, straight away I can tell it's mitosis, because if it's meiosis, the weight will not be the same as the parent cell, because after meiosis in that entire process, and that too, meiosis, it's not just one nuclear division like what's going on here, it'll be more than one. And also, it's not going to be the same as the parent cell because there'll be more number of cells. So that's not, so straight away we can say it's not meiosis. As one division only and it's returning to the same mass as the parent cell. These are the two important points. It's the same mass here. I'm just going to say it's mitosis and it's diploid because again only meiosis produces haploid daughter cells, so it's 32C. Okay. In a family, two parents have blood groups of A and B, and the three children have blood groups of B, B, and O. Okay, so we can, because it has O, one child has O, that is when both are IO, IO. And this can only happen when both parents have this present. Both parents have, so one has to be IA, IO, and the other has to be IAIO, other has to be IBIO. So IAIO, IBIO. So that's straight away that's 34C because this we need to have the recessive alleles both present. So for that to happen, both parents need to be carrying the recessive allele. So 33C is our answer. You can just do a monohybrid cross if you want to, but this is a more simple way. So which statement describes the type of variation? So continuous variation is usually caused by the environment only. No, it's caused by genetics factors and the environment. So it's not 34A. Continuous variation results in a limited range of phenotypes with no intermediates. No, it has a great range of phenotypes with intermediates. So again, 34B is wrong. Then discontinuous variation is usually caused by genes only. This is true. It's not controlled by the environment. So it's 34C. Let's look at the last option. Discontinuous variation results in a range of phenotypes between two extremes. No, this actually is a description for continuous variation. Discontinuous variation gives discrete phenotypes and there's no two extremes there. Okay, so hydrophytes, so like water lily plants growing in an abundance of water. So it shows a water lily plant which is a hydrophyte. So which statement explains why stomata are located on the upper surface of the leaf? So to allow carbon dioxide from the air to enter the leaf for use in photosynthesis. Okay. To allow carbon dioxide produced by photosynthesis. Okay, straight away this statement is not true. Carbon dioxide is not produced by photosynthesis. To allow oxygen from the air to enter the leaf for you. Okay, no, again oxygen is not used in photosynthesis. And allow oxygen from the water to enter the leaf for respiration. That's not true because it's on the upper surface of the leaf, so it's not actually facing towards the water, so that's not true. Also, that doesn't make much sense. The answer obviously is A here, and the more clear explanation is if you have stomata here, like these little blue dots I'm placing, if you have it down on the surface of the water, you're not actually going to get carbon dioxide. The plant won't be able to do photosynthesis. That's why it needs to be on the upper surface of the leaf so that it can actually get carbon dioxide via the atmosphere. So it's 35A. Okay. So which term is used when humans cross individuals with desirable features? So they're trying to get a desirable offspring. So obviously we can tell this is not natural. So it's not 36B. It's not 36C either. Because pure breeding is when you breed two things with the same characteristics, same alleles and everything. Pure breeding, you're trying to get a similar offspring. Genetic breeding is not the term here. Selective breeding is the obvious term because we are literally choosing organisms with desirable features. We're selecting organisms with desirable features. So evidently the answer is going to be 36B. Okay, 37, nitrogen cycle. So the nitrogen, what shows part of the nitrogen cycle? Okay. So nitrogen gas in atmosphere, nitrogen fixation, denitrification. So here denitrification is actually going to be the opposite arrow. It's going to be going from down to up, but this part is correct, this part is wrong, so A is wrong. B is correct because nitrogen gas in atmosphere, you need to make it into nitrate ions in the soil so plants can use it. And this is done by nitrogen fixation, so the arrow is facing the right way. And the nitrogen fixation can be done by fertilizers or lightning or other methods. And then denitrification is when you're removing those nitrate ions and making it into nitrogen gas in the atmosphere, which is done by bacteria in the soil. So denitrification is also the right faced arrow, so it's 37B. So let's just see why these are wrong. So nitrogen fixation cannot be the opposite way. It has to be, the arrow has to be like this. So that's why these two are wrong. And the denitrification arrow here is right, but here both are wrong. So 37B. Okay. So what is advantage of growing crops as a large-scale monoculture. So growing crops as a large-scale monoculture actually decreases biodiversity. So this is not true because you're planting one species, not multiple. So that's actually going to decrease your your biodiversity. Genetic variation is increased. That's also a false statement because again the same species, so it's not much genetic variation. Water use is increased. This is not an advantage. In fact, it's a disadvantage because scarcity, etc. So that's a disadvantage. Only 38D is answer. So the yield of the crop is increased. Farmers can make more money. So 38D is the obvious answer. So now we're moving on to the last two questions. Yeast can be produced using carbon dioxide and ethanol. What can be made using these products? Carbon dioxide, you can use it to make bread. We use it to make bread. It's what helps the bread rise. Biofuel and others are all fruit juice, that is pectin, and these are all enzyme related answers. Washing powder are also enzymes. Biofuel is ethanol. Ethanol is biofuel, and these are all enzyme related answers. And bread is carbon dioxide, so that leaves us 39D. Finally, we're on the last question. 40. Bacteria can be genetically modified to produce a human protein. What is the first step in this process? So we've already made a video about genetic modification. I made one earlier. Please go check that out after this if you want. So bacteria can be genetically modified. So what's the first step? So the first step we have is not insertion of human DNA into bacterial plasmid DNA. For that, first you have to isolate the DNA. It's not all of these. This is like the last step. The 40D is the last step. 40A is the third step. Fourth step. 40B is the fifth step. So the second and third step, here we have the first step actually. I completely messed up the way I said that. So first you need to isolate the DNA, making up the human gene. That is your first step. Then you cut the plasmid, leaving sticky ends. Then comes step A. So the step A will be this is actually the third step. Option B is the fourth step, and option D is like towards the last step. So that's it, guys. That's the entire paper 2 major in 2024 solved. Please consider liking and subscribing, and I'll see you in the next video. Thank you so much. Bye bye.